Chapter 14 goes over evolution and natural selection. We start by introducing Charles Darwin, who was born in 1809 and lived until 1882. He was an English naturalist who was the first to suggest an explanation for why evolution occurred. Charles Darwin's work on evolution challenged the established worldviews. Darwin proposed an idea which was a possible explanation for evolutionary change, which he termed natural selection. Darwin's hypothesis for evolutionary change, after a lot of testing, eventually became accepted as theory. And when I say theory here, I don't mean in the common term, I mean in the scientific term. In science, a theory is a carefully thought out explanation for observations of the natural world that has been constructed using the scientific method and tested heavily. Darwin voyaged from 1831 to 1836 on the HMS Beagle, which was the name of the ship. This ship had the goal of mapping the world's coastlines. And it's interesting, he actually only got a spot on the ship because in those times, captains were not allowed to fraternize with the crew. They believed that that led to mutiny. And so they would often have somebody like a philosopher or an educated person on board to kind of serve as entertainment for the captain. So they would have dinner together, they would have talks together, so the captain wouldn't be driven mad by isolation since he was not able to enjoy the company of the crew. So that's how Darwin got an invite to the ship and how he benefited and was able to observe all of the different places in the world and the different animals and insects that existed there. He observed many differences in the different places they went, and these observations played an important role in the development of his thoughts about the nature of life on Earth. This red line shows the path that the ship took around the world, and it's kind of mind-boggling that they were even able to accomplish this, given their total lack of technology at that time. But they followed this path, and they spent the majority of their time around South America and then here in the Galapagos Islands. Darwin made several observations that really helped to lead him to believe that that species evolve rather than remain the same. Whenever he left on the voyage, he was still a firm believer, like everyone else in the time, that we do not change throughout time. We were made the way that we are, and all of the variation that exists will stay that way. However, he kept observing different pieces of evidence that made him question that. The first of which was fossils of different extinct organisms that really resembled those of living organisms. Also, he noticed geographical patterns that suggested that organismal lineage could change gradually as individuals move into new habitats. And he observed that islands have diverse animals and plants that seem to be related, yet different from their mainland sources. One of the main things that he studied were the finches, which is this kind of bird that he found on the Galapagos Islands. He observed that although they were all very similar in shape and size and reproductive methods, they had very different beak sizes that had seemed to evolve to suit their food sources. He termed this descent with modification, the idea that they all descended from a common ancestor and were modified slightly to suit their different environments and their different food sources. Many organisms that he observed, like the sea turtles, he noticed that they would produce a lot more offspring than would actually survive. So even though hundreds or thousands of sea turtles will hatch on a given beach at any time, only about 10% of those turtles will grow to be adults. So only about 10% will actually survive. This is something he kept noticing with different species. He envisioned that the frequency of favorable characteristics would increase in a population through natural selection. This idea is that favorable characteristics are specific to a specific environment, and they could be favored in one environment, but not in another. Organisms whose characteristics are best suited to their particular environment will survive more often and they will leave more offspring, which means they'll be passing on those genes. Darwin had these tenets of evolution by natural selection, and usually this word is mainly used in religious or philosophy context, but considering he was alive in the 1800s, it makes sense that he used this terminology. So basically think of them as the main principles of natural selection. The first of which is that variation exists within a population. A population is not going to only express one kind of phenotype. Also, these variations are heritable, which means they can be inherited. The next tenet is that more offspring are going to be produced than can survive, and that's true for all species. And the next is that only those that are most suited to the conditions at that time are going to survive to reproduce. Individuals with advantageous variations are going to be more likely to survive and reproduce, which is going to lead to the gradual change in a population over time, which is essentially where the idea of survival of the fittest comes from, where beneficial traits are going to be passed on to future generations through reproduction. Whenever we say survival of the fittest, we don't mean the strongest or the biggest or the fastest or even necessarily the smartest. In this case, in evolutionary theory, the fitness is actually referring to organisms who have the right characteristics to survive better and leave more offspring. So these are the individuals who are best adapted to their specific environment. Darwin drafted his ideas into a manuscript in 1842, but he hesitated to publish them for 16 years. And it wasn't until another researcher who had read a lot of the same works that Darwin had read that led him to his eventual conclusions. He received a letter from Alfred Wallace, who basically outlined his theory of evolution by natural selection. So they had independently come to that same conclusion, that evolution must be taking place and that we must be naturally selecting genes to be passed on. Darwin was excited by this idea. He arranged for a joint presentation of their ideas in London. Unfortunately, that presentation never happened because the day before the presentation, Darwin's daughter actually drowned, and so they canceled the presentation. Obviously, Darwin was devastated, but he did later go on to publish his manuscript on the origin of species in 1859, so 16 years after he had completed that work. And he wasn't just kind of sitting idly during that time. He was doing a lot of other research and writing about other studies that he had been doing. So he just kind of let that one sit, and then was ignited by Alfred Wallace coming to the same conclusion and kind of emboldened to the point where he decided to publish. When he did publish, he ignited a huge controversy. Everybody was extremely upset with the idea that humans could have evolved from primates. There were a lot of satire cartoons that came out about him at this time, a lot of people calling him a heretic. But the scientific community soon accepted Darwin's arguments because the evidence was overwhelming. In his study of the finches, he cited that they are a closely related group of distinct species. All of the birds there are similar to each other except for the shape of their beaks, and genetic differences account for those physical differences that he observed in the beaks. For instance, birds with larger beaks, turns out they make more of a protein called BMP4 due to a gene that they have received for that increased production of that protein. There were various different beak sizes suited to various different things. So you can see here we've got our vegetarian tree finch, which is a bud eater. It has a kind of parrot-like bill. You can see that we've got this small and large insectivorous tree finch, which have kind of grasping bills, which are good for catching insects. This woodpecker finch actually has a beak that is able to perfectly hold small sticks and be able to fish out small insects from like holes in decomposing logs and trees. Interestingly, this sharp beak finch has a really sharp point at the end and was known to kind of sneak up on other birds and pierce their neck and drink their blood. Like, that's how it got the nickname of the vampire finch. And then we also have a small ground finch, a medium, and a large ground finch. You can see we've got a lot of variety in our beak shapes happening. Darwin supposed that the birds evolved from one single ancestor and then became kind of specialized in different particular foods based on their environment. Peter and Rosemary Grant also studied the medium ground finch on the island of Daphne Major in the Galapagos. And they did this, I believe they started in the 80s, and they did it over a period of about 20 years. They would go there and spend the whole summer there and study these finches, continuing the work of Darwin. They found that the finches preferred to feed on small, tender seeds, which were readily available in wet years where there was ample water in the environment. And the finches would switch to larger, harder to crack seeds whenever those small seeds became hard to find, like during years where there were drought and not enough water to produce a lot of the small, tender seeds. They found that beak depth increased when only large, tough seeds were available. So, like during those dry years. And here you can see a representation of their findings. Anytime they would have a dry year and suddenly there were limited supply of these seeds, they would notice a spike in offspring for having larger beak sizes, because suddenly it's advantageous for the species to survive if they have a larger beak and are able to crush those seeds that have all of the nutrients inside the shell. If their beaks were too small, they wouldn't be able to access the benefits inside the shell, and so that's why we were able to observe that difference. We also had one other researcher that went there between Darwin and the Grants, and he had opposite findings. So he had said that it doesn't matter what type of beak the finches have, they're all eating the same kinds of seeds. But it turns out he was there on a wet year, not this year specifically, but I'm just pointing to it because that's a wet year on this representation. And because he was there during a wet year, there was plenty of seeds anywhere the birds looked, so they weren't really in competition at that time. But Darwin and the Grants were both there long enough to observe what happened during the dry years, and that caused an increase in the bigger beak sizes. The Grants' work with medium ground finch is an example of evolution in action. And it's actually very interesting because a lot of times we think of evolution happening over hundreds of thousands or millions of years, but it turns out we can observe the effects of natural selection and evolution in real time in one lifespan by just spending enough time looking at a single species in a specific environment. So we know that the average beak depth increased after drought, and only those large-beaked birds were then able to crush the bigger seeds and actually survive to make the next generation, which is why that gene would be passed on. During the wet periods, there was a return of the smaller beaks in the next generation, and that's because there was plentiful small seeds and their offspring received the genes for smaller beaks because they were able to survive just as well as the finches with larger beaks. Darwin's finches on the Galapagos are a pretty perfect example of adaptive radiation. So this idea is just basically the idea of taking a single cluster of species and then observing the changes as it occupies a series of different habitats within a region. So you could imagine we drop one kind of bird in the close to the Galapagos Islands, and from there the offspring spread to adjacent areas with different kinds of habitats, and then develop different traits to suit those habitats. When we're talking about those habitats, we refer to them as niches. So in common day-to-day life, we know A niche is something that's kind of specialized. Maybe a niche interest is like a specialized interest in something. But in this case, we're talking about the habitats. So the specific niche that they are adapting to. And that's a specific habitat that they're interacting with different biological and physical challenges in the environment in order to survive. Each species that is subject to adaptive radiation will then evolve to become adapted to that specific niche that they find themselves in. There are many lines of evidence supporting Darwin's theory of evolution. One of the main lines of evidence is just fossil record. This is extremely direct, and we know fossils are the preserved remains, tracks, or just traces of once living organisms. They're created when organisms, or the tracks of them, are buried in sediment. And by dating the rocks in which the fossils occur, we can get an accurate idea of just how old those fossils are. Fossils in rock represent a history of evolutionary change, and fossils are treated as samples of data and are dated independently of what the samples look like. So in the beginning of fossil collection, it was really common to try and group every fossil that we found with the other fossils that it looked like. But we found that that's actually not the most accurate way to do things, because a lot of times we'll have very similar structures that develop independent of each other. And so the first thing that we do now is look at the date that those fossilized organisms would have existed, and then go on to sort based on their structure and different similarities that could indicate lineage and what ancestors they may be related to. But these successive changes through time are a data statement. They are a fact. They're not an opinion or a theory. We can plainly observe and can't deny that we are observing these changes to structures over time. Here's one example of those fossils. So the Titanothere, about 50 million years ago, the fossils had a barely perceptible bump in their nose. And as time went by, 45 million years, 40 million years, and then up to about 20 million years, we're seeing the development of changes for this defense structure that they have. Also, aside from just the skeletal anatomy, we have anatomical record that reflects evolutionary history in examples like embryos. So, for example, all vertebrate embryos do share a basic set of developmental instructions, and all of those embryos are still going to have a pharyngeal pouch, which is something that at one time would have developed into gills. Obviously, we don't have gills, but as embryos, we still all have the beginning makings of gills in that pharyngeal pouch. Also, all vertebrate embryos have this tail as well. When we're talking about these different structures, we've got homologous structures and analogous structures. The homologous structures are derived from the same body part that is present in an ancestor. It just could be modified and put to different uses in related species. So if we go to the next slide, you can see these are homologous structures. Basically, we've got the same general set of bones with modifications made for our different purposes. So you can see all of these in purple in the bat wing are the same bones that developed into the hand in humans and into the lower half of the leg in horses, and this portion of the fin in porpoises. And then same goes for these structures in gold, modified for their specific purposes. But they have a common ancestor and lineage. Analogous structures are structures that look similar, but actually have unrelated lineages. So these structures are actually the result of parallel evolutionary adaptations. And that makes sense. So we've got different organisms existing in different parts of the world, but the environments are similar, and similar enough that they're pushing for natural selection towards the same kinds of evolution at the same time, or at different times. So one example of that is bird wings and bat wings. even though they are so similar in structure, they were just developed in a parallel way. So independent of each other, not one coming from the other. This form of evolutionary change is referred to as convergent evolution. Also, traces of our evolutionary past are evident at the molecular level, not just in fossils. So we can kind of safely assume that organisms that are more distantly related, meaning they have more time between their development, should have accumulated a greater number of evolutionary differences than two species that are really closely related in time. The pattern of divergence can be seen at the DNA and protein levels when we look at those. So for instance, let's look at the number of amino acid differences between hemoglobin of these different vertebrate species and that of humans. So if humans are the base that we're looking at, there are about 8 amino acid differences between the hemoglobin of us and macaque monkeys. There are about 32 differences in our amino acids associated to hemoglobin and dogs. 45 between us and birds, 67 differences between us and frogs, and then 125 differences between us and lamprey, just only looking at the amino acids affecting hemoglobin. So you can see that is also confirmed. The further we go back in time and between descendants, the more differences we're observing. So it's like these evolutionary changes are stacking on top of each other. Also, the evolutionary changes accumulate at a constant rate in some of our proteins, which is very interesting. And that constancy is something we refer to as the molecular clock. So we expect the same kind of increase in evolutionary changes in our proteins. And that number or rate could be different between different proteins. There are a lot of critics over the years, less and less as time goes on, that have raised a variety of objections to Darwin's theory of evolution by natural selection. The objections have all either been refuted by biologists or just don't necessarily reject evolution, even though they're presented as if they do. One of the most common claims is the irreducible complexity fallacy. And this is usually argued by people who are proponents of intelligent design. And the argument is that the molecular machinery of the cell is irreducibly complex. However, natural selection is acting on the systems and not the parts, so that every stage of evolution, the system is functioning. So that's just basically to say all of these changes are building on each other. So it's not like we're having a grand design that needs to be all laid out all at once. We're making small changes to organelles in the cells, small changes to proteins, and then all of those changes are successive and building on each other. We're acting on the system as a whole. So one example of that is the mammalian blood clotting system, which has evolved in stages from much simpler systems over time. The book also talks about the Hardy-Weinberg equilibrium. And we know that variation within populations puzzled scientists for many years. One of the main questions was, why don't dominant alleles completely drive out the recessive alleles? Hardy and Weinberg in 1908 studied the allele frequencies in a gene pool and found that in a large population where there is random mating and the absence of forces that change those allele frequencies, like a dry year or a wet year or something that would drive selection, the original genotype proportions will remain constant from generation to generation. Because the proportions do not change, those genotypes are said to be in a Hardy-Weinberg equilibrium. When we think of equilibrium, we think of balance. So they're in a Hardy-Weinberg equilibrium, which is like a balance, where we're going to continue to have the same proportion of genotypes from one generation to the next. If the allele frequencies are not changing, that means the population is not evolving. Hardy and Weinberg arrived at their conclusion by analyzing the frequencies of those alleles in successive generations. And they looked at the frequency, which was the proportion of individuals that certain characteristics compared to the entire population. So basically, how many of these individuals have these certain characteristics compared to the entire population? Knowing the frequency of that phenotype for whatever characteristic, they could then calculate the frequency of the genotypes and alleles in that population. The Hardy-Weinberg equilibrium only works, though, if the following 5 assumptions are met. So first, we have to have a size of the population that is very large or effectively infinite. We can't just take a small sample of, 20 monkeys or 20 turtles and expect to meet this equilibrium, because we have a very specific selection at that point. Also, we have to assume that individuals are able to mate with one another at random. So we aren't having effects of artificial selection. All of the individuals are able to mate together freely. There's also the assumption that there would be no mutation taking place, and there could be no immigration or immigration. So immigration with an I is coming into an area. Immigration or immigration with an E is leaving that area. So you can't have a lot of the population coming and going and still consider this equation for equilibrium. Also, all alleles are replaced equally from generation to generation, and that indicates that natural selection is not occurring. Most human populations are very large and randomly mate with respect to most traits, and thus they are similar to an ideal population envisioned by Hardy and Weinberg. For example, the frequency of heterozygote carriers for recessive genetic disorders can be estimated using the Hardy-Weinberg equilibrium. We have 5 factors that can alter the proportions of our homozygotes versus our heterozygotes, enough to produce significant deviations from those predictions. And these are the main factors that would influence that. So first is mutation, next is non-random mating, then genetic drift, migration, and selection. Mutation, as we know, is a change in a nucleotide sequence. So like how we can see here, UV light has caused the nucleotide that should have been thymine to be replaced with adenine. Mutation rates are going to be generally too low to significantly alter the Hardy-Weinberg proportions. And mutations will have to affect the DNA of those germline cells, or the mutation is not going to be passed on to offspring. However, we all know that no matter how rare a mutation is, it is also the ultimate source of all genetic variation that's happening in a population. When we talk about non-random mating, it occurs when individuals with certain genotypes will mate with one another more or less commonly than would be expected by chance. So sexual selection is choosing a mate, often based on physical characteristics. If it's not random, then that means there's something causing it to be more, causing one group within the population to be more likely or less likely to mate with another group. Non-random mating alters the genotype frequencies, but it won't alter the allele frequencies because we're still passing on those alleles. It's just whether or not they're going to be expressed, or whether we'll have the right genotype for them to be expressed. Also, genetic drift. When we talk about genetic drift, it's just describing random changes in allele frequencies. And in small populations, the frequencies of those alleles could be changed drastically by chance alone. In extreme cases, individual alleles of a given gene could be only represented in very few individuals, or they could also just accidentally be lost if individuals fail to reproduce or die. You can see an example of genetic drift over here. Imagine that we've got a two different phenotypic expressions for this species of bird on the mainland, and imagine that only a small group of those birds would migrate to an island nearby, and suddenly we have a shift in the allele frequencies in the overall population as these birds on the island reproduce. So they're carrying only the allele for the red feather expression, whereas the yellow feathers are left on the mainland. Now we have a portion of our population that is representing just a few of those alleles in the overall population. And if you consider that all of these birds possibly didn't make it, the conditions were too harsh to survive, we could end up losing all of the individuals in that group, and therefore the alleles that they carry for that red feather expression. A series of small populations that are isolated from one another could come to differ strongly as the result of genetic drift like this. So this is how we see divergence in different characteristics of the same species that could eventually lead to speciation. But the founder effect is what occurs when we have just one of a few individuals that will migrate and become the founders of a new isolated population that is some distance away from the original place that the population existed. The alleles that they carry will become a significant fraction of the new population's genetic endowment, or their genetic inheritance, the genes they are inheriting. And the founder effect is important in the evolution of organisms, especially on oceanic islands, because they're so reproductively isolated. Another example of that, aside from what you could imagine of those birds being the founders of that population on that island in the previous slide, we also have an example of with humans in the Amish community. So in the 1800s, one of the founders of the Amish community in North America carried a gene for this disorder that results in your offspring having extremely shortened limbs. So arms and legs are very short. And this is still more prevalent in the Amish community than any other population in the world today. And that's because of their very limited pool of genes for reproduction. So they're not trying to mate outside of their community, which really limits the options of new alleles to be introduced that could possibly combine and interact or mask the expression of that disorder by having more individuals that are either heterozygous for that allele or don't have the allele at all, which would create more of a balance and kind of dilute that genetic pool. But as a result, we've got a shift in the frequency of that allele in this population compared to the general population of humans. Another form of genetic drift is caused by a drastic reduction in the number of individuals in a population, and we observe the bottleneck effect when that happens. This occurs when a natural or human-caused event, something like a hurricane, a fire, oil spill, some really big natural disaster, a really big tragic event that causes loss of the majority of the individuals in a given population of a species. In this case, the survivors are just going to be a very random sample of genetics. It's not really determined by who's fittest to survive. It's just completely random based on who was able to survive this catastrophic event. And that means we're going to have a random sample of the genetics for that population that's going to continue to be passed on. The allele frequencies of the population are going to reflect only those individuals who are able to survive and go on to mate. So here we can see what the parent population looks like. If you could imagine, these are the genes that they are carrying. When we have a catastrophic event and all of these members of that population die, we're left with a random sample of the surviving individuals and therefore a random sample of the genetic information that can be passed on. So then our next generation ends up being modified from what the original population's genetic makeup was. And this is the bottleneck effect. Also, migration is just referring to the movement of individuals between populations. So the movement of individuals can be a really powerful force upsetting the genetic stability of natural populations. And the magnitude of those effects is going to be based on two different factors. First, the number of migrants in the population compared to the original population. And second, the difference in allele frequencies between the migrants and the original population. So, did only a specific set of genes make it out of that population in this migration, or what was the frequency of the alleles that were carried out compared to how much there are in the original population? Those are the things that could affect this. When we talk about selection, according to Darwin, it occurs if some individuals, due to their inherited characteristics, are going to end up leaving behind more progeny than others. And this can either happen by artificial selection or natural selection. So in artificial selection, we have a breeder that is selecting those desired characteristics. So just like when we're breeding specific breeds of dogs or horses or sheep or whatever we're intentionally breeding, we're choosing the desired characteristics we want, and that's how we're making the decision about which members get to reproduce. So that's artificial selection because someone is making the choice for who gets to reproduce. In natural selection, that is led by the conditions in nature and the environment, which is going to determine which individuals are the most fit to reproduce and the most fit to survive. When we talk about natural selection, there are 3 types of natural selection that we tend to see. The first is stabilizing selection, then disruptive selection, and directional selection. In stabilizing selection, so first let's look at this example. We've got body size and the number of individuals that have that body size. In a stabilizing selection, it turns out we are getting, so if this is the original population and the original kind of frequency of size, then as natural selection goes on, we're going to observe that peak get narrower and narrower towards the middle of our range of possibilities. And that means it's stabilizing. So in this instance, it's not beneficial to be smaller than this range. It's not really beneficial to be larger than this range. The main benefit for survival is within this weight range for this species. So we see a narrower peak. We're getting rid of the extremes of the options for this expression. Disruptive selection is kind of the opposite of that. So in that, we're favoring the extremes. Here you can see there's no benefit to being middle-sized, but there is a benefit to being both smaller and larger in this species. And that's how we end up with 2 different peaks, because it's not as beneficial to be this weight, but it could be beneficial to be smaller or larger based on the environment in that specific scenario. So here we're favoring the middle. Here we're favoring the extremes. And then in a directional selection, we're favoring one of 2 sides. So in this case, you can see it's not beneficial to be smaller than about 90 grams for this individual. But we're going to see that peak start to shift towards the larger end of things. And because we're moving our peak in a specific direction, this is directional selection. So in this instance, it's only beneficial to be larger. That is the benefit, and those are the genes that are being selected. When we talk about stabilizing selection, a great example of that is human birth weight. So this is an example of a stabilizing selection. You are most likely to survive as an infant if you're born within this weight range. We notice increased mortality rates under that weight range and increased mortality rates if we are larger than this weight range. So that means we're getting rid of the extremes and favoring the allele that is going to produce this kind of range of birth weights in the middle. When we talk about disruptive selection, this is one example of that. So the light coat color in this pocket mouse is going to make this mouse vulnerable if it hangs out in the habitat of the lava rocks. The dark coat color of this mouse is going to allow it to blend into the lava rocks, but it would stand out a lot if it were out here in this sandy environment. So as a result, we've got an example of disruptive selection because it's going to benefit the mice to either be really dark or to be light and sandy colored. But if they were to come out with a color that is kind of in between these 2 colors, suddenly they're going to stand out on lava rocks and in the sand and be really easy prey for predators like this owl. So that's why it's a benefit of the extremes. So we've got the darker expression on this side, and then the other would be the sandy colored expression for this mouse. Another example of disruptive selection are the finch birds that we talked about. So the finch have a selection for either this large seed or this small seed, which means they have a selection for either the large beak or a small beak. A medium beak in that middle range is not going to benefit them as much because it'll be too small to crack the large seeds. It'll be too big to reach in the small crevices where they could find these small seeds. So that's why we are favoring both sides, but not the average in the middle of that range. Directional selection, our example is fruit flies. So we notice in our original population of fruit flies a pretty significant tendency to fly towards the light. But there's also a gene that indicates they will be more likely to fly away from the light. And what we notice is throughout the number of generations as we watch these species reproduce, a less and less affinity for that desire to fly towards the light. Now we have far less of the fruit flies that are going to fly towards the light, and the majority of them are going to try to avoid the light, which would benefit their survival because they're going to be easier to be seen for predators in the light and safer if they avoid it. We discussed sickle cell disease, which is a hereditary disease affecting hemoglobin in the blood. And this disorder results from a single nucleotide change in the gene that encodes beta hemoglobin. It's basically going to change that 6th amino acid in the chain from a very polar glutamic acid to valine, which is nonpolar. As a result of that, the hemoglobin molecules are going to clump together and deform the red blood cell into a sickle shape, like we see here. Here's that sickle shape. So, as a result, our red blood cells are not going to be able to carry as much oxygen, and now we have red blood cells that are very likely to form clots. People that are homozygous for the sickle cell genetic mutation are frequently going to have a reduced lifespan because of that. Their red blood cells are sickled, they don't flow smoothly through the capillaries, and then we have reduced oxygen transport as a result. But individuals that are heterozygous will still make enough functional hemoglobin to keep their red blood cells healthy, even if there are some sickled cells in the mix. The frequency of the sickle cell allele is about 0.12 in Central Africa. About 1 in 100 people is homozygous for the defective allele and develops the fatal disorder. Sickle cell anemia strikes about 2 African Americans out of every 1,000. If natural selection drives evolution, the question is, why has natural selection not acted against that defective allele in Africa and just eliminated it from the population there? Why does it even still exist? Why is it still expressed? The answer is heterozygote advantage. So we talked about in the previous chapter how if you do have a single allele for sickle cell, which means you can make enough red blood cells to get by and have decent oxygen transport and a pretty normal lifespan, if you have one single allele for sickle cell, you also have an advantage in defending against malaria. So that is something that we found. The payoff in survival of the heterozygotes in this case is making up for the price of death in the homozygotes of this general population. So even though if you get both alleles, you're likely to have a shortened lifespan and have negative impacts, if you have a single allele, there is an advantage in the way that you're able to fight off malarial disease. The stabilizing selection in this instance is occurring because of that malarial resistance. This is just another example of that map that we looked at where we could see the sickle cell allele in Africa, how common it is compared to the widespread effect of malaria in Africa. In the very beginning of the semester, we also mentioned our peppered moths and the effects of industrial melanism on them. So these are the moths that used to be kind of that white and black peppered color. And then as the Industrial Revolution came about and we're increasing our production in industry in many ways, now suddenly we were noticing more and more moths coming up with this increased melanin pigment. And they were showing up in darker forms more and more often because it allowed them to camouflage with the soot that was covering everything from all of the industry operations and all of the air pollution. But what we've noticed over time is as we've been reducing our air pollution, there's been a reversal of that industrial melanism in the peppered moths. And it also seems to be occurring in England. So you can see we used to have about 93% of our moths would be born with that melanic gene expression, so they would be very dark. As time goes on and air pollution has been reduced, this is the number of melanated moths that we were observing in 1995. So you can see we had a severe decrease as we started to address air pollution. Another great example of selection is guppies. So this is something that we were able to observe in the wild and also recreate in the lab. But guppies are a fish that are really common in Trinidad. They're found in two different stream environments, one upstream of waterfalls and one downstream of waterfalls. The pools that are above the waterfalls have guppies that live with the killifish, which is not really a predator of the guppies. You could think of them getting along and being pretty equal. However, the pools below the waterfalls have guppies that live with pike cichlids, which are voracious predators of guppies. So they see the guppies as a snack. The guppies are trying to survive. How does this affect the way that the genes are expressed in these different populations of guppies? Well, it turns out the guppies that are in those high predation pools with the pike cichlids trying to eat them are not as colorful as the guppies in the low predation pools. And it also turns out they're smaller and they reproduce way earlier than the guppies in the safer pool. So these differences are suggesting that function of natural selection, where if you're going to live in a population that has a high risk of predation, then it's more beneficial to be smaller, to be less noticeable, so have muted colors and less spots, and to reproduce sooner because your lifespan is probably going to be shorter or it is at greater risk of harm. Here's what that looks like. Here's our guppies in our upper pool with the killifish who do not eat them. These guppies are going to be brighter, they have more spots, they're larger. That's really good for attracting mates. So that does serve a benefit in this specific environment. However, these guppies are subject to predation from the pike cichlid. They're trying to hide and survive and pass their genes on before they die, so that means they're smaller. They have more muted colors so they can be more camouflaged with their environment, and they will reproduce more quickly than the guppies upstream. John Endler conducted experiments trying to replicate this observation, and in a controlled lab setting, he basically just kind of replicated what was happening. He had three conditions: one where there was no predator present, one where there was just killifish present with a low predation risk, and one where there was cichlids present, which has a high predation risk. After just 10 generations, the guppies from the no or low predation risk pools were both larger and more colorful than the guppies that were in that high predation pool. He later found the same results in field experiments. So we observe the less predation there is, the brighter colors, the more spots, and the larger the guppies will be. When we talk about speciation, it's just the macroevolutionary process of forming new species from pre-existing species. So you could think of this as the moment where we have one species that has two groups that keep getting more and more different characteristics. So whatever their different habitats are, we're favoring different genes. And as a result, they're getting more different as time goes on until the point where they have become so specialized and become different enough that now natural selection may act to keep them that way and make it to where they can't breed and create viable offspring. So you could think of speciation as the moment where one species diverges into two. Ernst Mayr in 1942 coined the biological species concept, and that defines a species as groups of actually or potentially interbreeding natural populations which are reproductively isolated from other such groups. So populations whose members do not mate with each other and cannot produce fertile offspring are said to be reproductively isolated, and therefore members of a different species. When we talk about barriers to reproduction, they are called reproductive isolating mechanisms because they're isolating the pool that is able to reproduce within a population. This causes reproductive isolation by preventing genetic exchange between species. And we've got two mechanisms for that. We've got pre-zygotic isolating mechanisms and post-zygotic isolating mechanisms. Don't let this word trip you up. If you just think of the moment a zygote is formed, these isolating mechanisms are happening before the zygote could even be formed. So something to prevent reproduction before there's even a chance for the sperm to meet the egg and form a zygote. Post-zygotic isolating mechanisms are ones that will prevent the proper functioning of those zygotes. So they were able to, two organisms were able to mate together and produce a zygote, but something is going to prevent that zygote from becoming a viable adult that's able to produce offspring. There are 6 different pre-zygotic isolating mechanisms, and they're seen here. So we've got geographical isolation, ecological isolation, temporal isolation, behavioral, and mechanical isolation. And then also just the prevention of gamete fusion, where the sperm does meet the egg, but they're not able to fuse together to become a viable embryo. When we talk about post-zygotic isolating mechanisms, we're really just talking about hybrid inviability or infertility. So basically we made a zygote, but that zygote is not going to become a viable embryo or a fertile adult that's able to pass on the genes for that species. When we talk about geographic isolation, it means we have species that are just in different areas. So even though they may be able to mate if they could find each other, they're just separated by some physical barrier. It could be a river, could be a mountain range, could be the ocean, but they cannot reach each other to mate, and so they're geographically isolated. Ecological isolation is similar, but slightly different. So in this case, species occur in the same area, but they occupy different habitats within that area. And as a result, the survival of hybrids is going to be very low because those hybrid individuals that are born are not going to have the genes to adapt to either of the environments of their parents. In temporal isolation, that means that the species could potentially reproduce, except for the fact that their reproduction cycles happen maybe in different seasons or just different times of day. So they're basically, they're not able to reproduce at the same time, and therefore they're not going to be able to reproduce. Behavioral isolation is when species won't choose each other because of a difference in their mating rituals. So this could be like the birds that are listening for a very specific song in a mating call, and even though physically they could reproduce with another kind of bird, they won't choose to because they don't present the right mating ritual or the right bird song. This also is an example, there's a similar situation with crickets, where crickets have a very specific song that they're listening for in their mating ritual. Mechanical isolation is an instance where there are structural differences, like physical differences, between the two species that's going to prevent them from being able to mate together. And then prevention of gamete fusion is where we have sperm that has found the egg, but is not going to be able to fuse to form that zygote. For our post-zygotic isolating mechanisms, we've got hybrid inviability or infertility, And in this instance, hybrid embryos do not develop properly. So the hybrid adults don't survive in nature, or the hybrid adults will survive for a certain amount of time but not be sterile, but they will be sterile, or just have reduced fertility, so they can't pass on those genes. So when we talk about ecologically isolated species, and one example of that is lions and tigers. Technically, they could travel to each other and mate to produce offspring like this liger here, but that's not going to be possible because they're just not looking for each other in those separate habitats. Let's see. So yeah, we've got our temporal isolation and behavioral isolation. You can come back for these slides of the descriptions. They're basically the same thing we went over in that first table. But when we talk about our post-zygotic isolating mechanisms, it basically means if hybrid mating does occur and the zygotes are produced, a lot of those post-zygotic factors are going to prevent the zygotes from developing into normal individuals. In this instance, we've got hybrids where the genetic complements of two different species could be so different that they can't function together normally in the embryonic development. And in this case, even if the hybrids do survive that embryo stage, they're not going to develop normally. Or maybe they'll develop to adulthood, but those hybrid organisms will be sterile and not be able to produce viable offspring of their own. So here are some examples of that with temporal isolation, where we're separated by time, the timing of our reproductive cycles. We've got an example of frogs in ponds. So there are some frogs in ponds that exist with other species of frog where potentially they could mate, but because one kind of frog mates in the day and one mates in the evening, they're not going to kind of sync up to be able to reproduce. Ecological isolation, we've got that example of our lion and tiger, which could potentially mate together, but typically don't because of their differing habitats. Our behavioral isolation has to do with the mating rituals, like the song of the cricket, where a cricket is listening for a specific song, and even though they physically could reproduce with another species of cricket, they won't because they don't match up behaviorally. Our example of mechanical isolation is bush babies, which have genitals that are positioned in such a way where the male cannot often reach the female in order to reproduce. So they have a physical barrier. They physically cannot reach each other with their genitalia and therefore are not able to reproduce. Our post-zygotic barriers we're talking about, there's examples of the hybrid inviability between different species of frogs. Also hybrid infertility. So we observe that with horses and donkeys. They are able to mate and produce a mule, which is a hardworking animal that is very physically capable, but that mule is going to be infertile. It will not be able to then reproduce and pass on the genetic information any further. And then when we've got hybrid breakdown, that's an example where maybe the first generation was successful, but the second generation is going to produce all infertile organisms within that species. So here's a great table to summarize all of those differences. I would recommend using this when you're going over your study guide. We can see the examples of temporal isolation, ecological, behavioral, and mechanical. And then here are our examples of hybrid inviability, infertility, and breakdown. That's all of chapter 14. Now we're going to go on to chapter 15, which is all about classification.